Okay, students. So, we are going to start off the lesson with the first activity. What are we going to do now? We are going to observe the cells present in different types of plant parts. So, for that, I have a beetle leaf. We will be observing the lower epidermis of the beetle leaf. Then, we will need to observe a very thin piece of the potato tuber to observe the cells present in it. And the third specimen, it is a balsam plant. It has a very thin stem, so we will be observing the cross section of this stem. I will start off the activity by preparing a temporary slide of the beetle leaf lower epidermis tissue. So for that, we will have to take the beetle leaf, and you all know this is the upper epidermis and this is the lower epidermis. So here we use the lower epidermis because there are more stomata present in the lower epidermis. We can take thin peels by cutting very thin pieces. So I can do that as well. Here we will have to cut very thin peels first. For that we have to use a very sharp blade so that you can get it as thin as possible. And once you cut off small thin peels, you have to put it into a watch glass containing water. It can be any glass container containing water. Then I have the brush to transfer the specimen onto the glass slide and the cover slip to cover the slide. So first I will try to get the thin peels. Like this we can cut thin peels. We have to make sure it has to be a thin layer and also it has to be a whitish color layer. If it is too thick or green in color, then of course there can be many layers of cells that will not be very clear to you. We can do it this way, or there is another method. Here what I have done is I have taken a piece of the leaf, and if you tear it like this, also you can get very thin peels, and from that you can see it's a very thin layer. I can cut off the thin peel, and that also can be used as the tissue specimen. Then what do? We have to take a clean glass slide. I have one here now, and then using a paintbrush I will transfer the thinnest specimen from water. Before that, I will put a drop of water on the glass slide. You all know why? So that the tissue doesn't dry off. It will be fresh and we will be able to observe the parts of it or the cells present more clearly. And then I'll transfer the thinnest specimen. Since it's a slightly large specimen, I will put another drop of water, and on that I will be placing a cover slip. Have to take out the cover slip. It's a very thin cover slip. So what do we have to consider here? We should make sure that there are no air bubbles trapped inside. So for that, I will place the cover slip like this and drop it onto the specimen. Then of course, there will be no air bubbles trapped inside. Now the temporary slide is ready for observation. So now we have to check whether the microscope is ready. You can see the light is on, so there is enough light falling onto the stage. You can observe the specimen clearly. Then I will make sure that we select the low power, and also I will place the slide prepared by me on the stage and adjust it so that the specimen comes into the correct position. Okay, now it's all set. Then we will observe the tissues that are present in the lower epidermis of the beetle leaf. Okay, so first I will adjust the coarse adjustment knob and see. Now students, you can see the cells present in the lower epidermis. Can you all see dark color like oval shape, dark spaces? You all know those are the stomata. And around that you can see two bean-shaped cells. Those are the guard cells, and also the rest of the cells are different in shape and also they are green in color. So these are the different types of cells present in the lower epidermis. So students, that was the observation through the low power. You all saw the stomata, the guard cells and the different epidermal cells. Now to observe it more clearly, I will change the power to a higher power. Now I am changing it to a higher power and now we will observe the specimen again. If I do a little bit of adjustment, you can now see it clearly. The cells, the shape of the cells are more clear to you. You can see the greenish color cells and also you can see the different shapes. The shape of the epidermal cell as well as the shape of the guard cell. In the middle of the guard cells, you can see the stomata clearly. So students, you have observed all the different types of cells that are present in the lower epidermis. And you can identify the different known cells. Okay, so that is the first plant part that we are going to observe. Then now I'll move on to the second plant part, that is a potato tube. So now I have cut a small piece of the potato tuber. This is the part that we are going to observe. So from this part again, I will have to peel out or cut out very thin layers. So here you will have to cut very thin layers. You know the reason why we need to cut very thin layers. Thin layers will give you less number of layers of cells so that it will be very easy for you to observe it. And as we cut off the layers, we have to put it into a watch glass containing water like we did before. I'll cut out few more pieces so that we can select the thinnest piece from the pieces that we have cut out. Okay, now I have cut a few pieces there and I have the specimens from the potato tuber ready. Then I need to take a clean glass slide and onto that I have to put a drop of water. You all are very familiar with preparing temporary slides. From smaller grades you have done this. So therefore I am not going into detail about the preparation. And from this I select the thinnest piece and there is enough water. Onto that I am going to place a cover slip. That is also glass. And what is the consideration here? We have to make sure that there are no air bubbles trapped inside. So for that we hold the cover slip slant like this and place it on the specimen. Now I have prepared the temporary slide containing a thin layer of potato tube. Now, I will have to remove the slide that I placed earlier. This is the beetle leaf. I place the new slide I prepared. Okay, again I have to make sure the specimen is in the correct position. That's correct. And also I have to adjust the lens to the low power. Low power is ready. Now I have to observe it. So first I will observe it through the low power, and with a little bit of adjustment you can see, students, clearly the shape of the cells. You can see almost all the cells are spherical in shape and they are all similar in size. That is the observation you can get from this slide. All the cells of the tissue are similar. So that is not clear enough. Then what do? I will change it to the higher power so that we can get a more clear observation. Now with this higher power, when I do a little bit of adjustment, now you can see more clearly the spherical shaped cells, and they are almost the same size, and there are spaces in between the cells, and these cells all look very similar to each other. So students, you can get the observation clearly how the cells of the potato tube look like. It was different from the first slide we prepared, that is the lower epidermis of the beetle leaf. Now again, I will be using the third type of plant part, that is a stem. And we will see how that will differ from these first two slides. So I will remove this slide. I am going to use another slide. I have to prepare it first. So here you can see, students, this is a balsam plant. Now we usually take this balsam plant for the evenness and the thin skin, and also because of the color you can observe the cells clearly. So here you can see this part is a little darker in color, this part is slightly light in color. I will be cutting very thin cross sections from the darker part of the stem. So first I will have to cut out small specimens. They have to be very thin. And again, to make sure that we get very thin pieces, I will cut more than one layers. Here you can see I am getting very thin layers. Those are the cross sections of the balsam plant stem. I will cut one more. Right. So now here you can see it's all colored. So here itself you can observe slightly the different regions. When we observe this through the microscope, that will be very, very clear to you. So now I am going to make the slide again a clean glass slide. I will put a drop of water. I might need a larger drop if I am using a very thin layer. I will take the thin layer and transfer it onto the glass slide. So here the specimen is there and there is enough water, so I have to put a cover slip onto that. So for that, I have to make sure that there are no air bubbles trapped inside. I hold it slant and place it on the specimen. Now the temporary slide mount of the cross section of the balsam plant stem is ready. I will first adjust the power to low power, then place the glass slide, that is the temporary slide, and make sure that the specimen is in the correct position with enough light falling on it, and now it's ready to observe. If I adjust it and observe, you can see the cells are reddish brown in color because of the color of the stem. So you can identify the different cells. Here you can see in the middle part of the stem there are very tiny cells, whereas when you go towards the outside the size of the cell increases, and towards the periphery you can see oval shaped dark color regions. Those are the transport tissue, that is the vascular bundle containing the different types of cells. We will discuss that later. For now, you can observe the difference in the size and the shape of the cell. So you have observed 3 different parts from 3 different plants, and you have seen that there are different types of cells which exist together to form the tissues of these parts. And when you saw, the beetle leaf had green colored cells, whereas the potato tuber had identical spherical shaped cells which looked similar. And the stem of a balsam plant had different types of cells present in it. But there were many similar cells. That is what we call as the tissue. So from all these observations, you all can understand that different cells are there. They have shapes and sizes, and similar cells together forms a tissue. From the first activity, you know how tissues look like. And you know what tissues are? A group of cells that have the common origin and that are adapted to perform a certain function. So now we will be looking at a plant part. For that I will be using a rampe root. And we will use this to see how the external view is different based on the type of tissues that are present in the plant. So here you can see this is a rampe plant. There is a very large plant here out of the rampe plant, so that you can see the parts clearly. And in this you can see all these stilt roots. The main root is there in the middle, and there are the stilt roots that start from the base of the plant. That is to support the plant to stay erect in the soil, which is a muddy, loose soil. So we'll be observing the stilt root. Here you can see there are different stilt roots, and this is a clear root that you can observe. And here you can see the shape. It's broader and almost the same diameter, but towards the end of the or the tip of the root, you can see it becomes a little more thinner and gets pointed at the end. And you can observe a color change. Here it is a darker color, almost like a brownish color, but when you go towards the tip of the root, you can see the color becomes lighter and green, and this is a light yellowish green in color. And here also you can observe the different roots with the different color shape. What is the reason for this color change? Here there are cells inside that are mature. So because of that, they are more stronger and also darker in color. But here there are cells which are dividing and somewhat immature. That is why you see the lighter color. So from this observation, you can understand, depending on the type of cells, the part of the plant shows different color and structure. So then only the cells can do their function, or the cells that make up the tissue can carry out their relevant function. That is the observation of this activity. First, we observe the nature of the prop root of a rampe plant. Now, we will get a longitudinal section of the part of the root and then observe it in the microscope. For that, I will select one of the roots which shows a color difference. So, for that, I can select this root and I'll be cutting a small piece from that root. Okay, so I won't be needing the plant anymore. Here I have the part of the root. Here also you can observe the slight difference in color. Then we will have to cut a longitudinal section. So I have to cut it through this root. For this, I have to use a very sharp blade so that it does not damage the cells. And from this, again, I will need to take a very thin layer. And we have to make sure that we get the long, thin layer starting from the point till we get to the tip. So I have to be careful when I cut the longitudinal section. Again, I will take another. I will make it a bit shorter and take another section. Now I have got a thin layer. So the next step, you all already know, we have to take a clean glass slide, which I have it here. And on that, I will use the blade to place the section. So I have the part of the root here. This is a longitudinal section, and since it's a larger part, I am not going to use a cover slip. So, but I have to make sure that the specimen does not come in contact with the lens of the microscope. So, now I am going to observe it under low power. I'll start from the base of the root, that is the part where we cut it off from the root. So, if we look at the cells there, you can see, students, the cells are in the middle. You can see many layers of cells, and the cells are very much identical or similar in shape and size. And we will see how the size changes when we go from the base to the tip of the root, and also towards the outer line of or the periphery of the root. You can see that there is a darker color region. Now I will move the specimen towards the tip so you can observe it clearly. Now observe the size and the color change in the cells present. Can you all see that the color is becoming darker and the cells are becoming smaller? So students, in this you can observe that towards the base, that is the part where it's connected to the root, the cells were larger and lighter in color. And when we go towards the tip of the root, the cells become darker and smaller in size. So with that observation, you can understand even in one part of the plant, there are different types of tissues. The tissues that are dark and small in size are the meristematic tissue. And the tissues which are more larger and which have a distinct shape and lighter color are the permanent tissues. We are going to do this activity to observe the different types of tissues present in a cross section of a plant stem. So for that, we can either use a pumpkin stem or a tridac's stem. Here I have a pumpkin stem. It's only a part of the stem. From this, I will be cutting very thin cross sections to observe using the temporary slide. So you know the procedure. We have to cut very thin layers of the cross section, and also as I cut the cross sections, I have to put it into water so that it doesn't dry off quickly. So now I have taken 2 cross sections. I will take a few more so that we can select the thinnest one. Right. So I have a few thin cross sections of the pumpkin stem. So what do I do next? I need to take a clean glass slide, and onto that I put a drop of water, 1 or more drops, so that the specimen will not dry off. Then from the layers I have cut out, I will select the thinnest one and place it here. There is enough water, and on that I have to place a cover slip. I will place the cover slip onto that slide. And what do I have to consider? When placing the cover slip, I have to make sure that air bubbles are not trapped inside. So now the temporary slide is ready for observation. I will place it on the stage as we normally do, and make sure that the specimen is in correct place and there is enough light falling on the specimen. Okay. So first, what is the power that we have to use? It is the low power. So through the low power, we can observe the full area and see how the cells look like. So here, when I do a little bit of adjustment, you can see, students, there are different layers of cells. In the middle, you can see different shape and different size, whereas towards the periphery, you can see different size and shape. And also, you can see the darker regions, which are the vascular bundles or the transport tissue, which contains the xylem and phloem, which we will be discussing later. So this is under the low power. You have already observed this before. What we are going to do now is, we are going to observe a small region across under high power to differentiate the cells more clearly. So I will change to high power, then I do the slight adjustment. Here you can observe the size and the shape of the cells. Towards the middle, you can see there are very tiny cells. But when you go towards the outer circumference of the stem, you can see the size of the cells increasing and the shape is more prominent. You can see polygonal shaped cells. And also in between, you can see the darker regions. You can see that clearly, students, the darker regions, that is the xylem and the phloem. in between the xylem and the phloem, where you have the middle line, those cells look very small and you can identify that region. So that is the meristematic tissue. So the rest of the cells that you are able to identify are permanent tissues, and the cells in the middle, which is the cambium, contains the meristematic tissue. So from this observation, students, we have identified the two main types of cells based on cell division. The cells that divide all the time, or the main purpose of the cell is to divide, are the meristematic tissue. And that was the cambium layer you saw between the dark regions. That is the xylem and the phloem. And the rest of the cells that you saw in different shape and size, especially the polygonal shaped cells, they come under the permanent tissue category. So with this observation, you all now have an understanding about the two types of tissues, meristematic and permanent tissue. Now we are going to observe the permanent slide of a parenchyma tissue. You know the features of parenchyma. What are they? The cells are isodiametric. That means they are spherical in shape. And also they have prominent intercellular spaces, and also they are living cells. So to observe this, I will use a permanent slide. Okay. I have this permanent slide of the parenchyma tissue, which I will place it on the stage. I make sure the light is on, there is enough light, and we have to move the slide to adjust. Okay, the specimen is in place on the stage. So first I will observe it through low power. Already the low power lens has been selected. Let us observe the parenchyma tissue. Now you can see, students, the parenchyma tissue, you can see the isodiametric shape, and also the cells are all similar to each other. We can actually say they are identical to each other. And you can see the intercellular spaces. So those are the features that you can see under higher power. So students, these are the observations that you can get by observing these permanent slides. Now we are going to look at the second type of simple permanent tissue, that is the collenchyma tissue. We have already discussed the features and you know how this differs from the first type, parenchyma. So now I'll select the tissue that is relevant to this. Okay, this is the permanent slide that has the collenchyma tissue. So again, I will mount it on the stage. Now I make sure it's the low power lens, and also there is enough light falling on the specimen. Now the slide is ready for observation. I will observe it under low power. Here, students, you can observe that the cells are polygonal in shape. And also you can see that the corners of the cells are thick. You all know that it is cellulose. So these are the main observations that you can see from this slide. Now that we observed the collenchyma, you can remember the parenchyma tissue, and you can now differentiate between the two tissues. Now we will observe the third type of simple permanent tissue, that is the sclerenchyma. So for that, I will first select the tissue. Okay, right, that has the sclerenchyma tissue here. So I will place the permanent slide, and also I make sure that it is in the correct position and enough light is falling on it. And first we will observe it through the low power. Now when I do the adjustment, coarse adjustment, you can see clearly the cells that are present in sclerenchyma. You can see the evenly thickened cell walls and the lumen present inside. And also the shape is polygonal. There are polygonal shaped cells with the thick cell wall and the lumen present. That is what you can see through the low power. To observe it a little more clearly, we will now observe it through the high power. So here I will use the high power to show you all the sclerenchyma tissue. And when I do the fine adjustment, you can see it more clearly. The polygonal shape of the cell, the sclerenchyma cells which have the thick lignin cell wall and the lumen in the middle. So that is a distinct feature which you can use to differentiate sclerenchyma from the other two types, that is parenchyma and collenchyma. So with these students, we have observed all three simple permanent tissues, and you will be able to differentiate and identify them separately. Now we are going to observe the two types of complex permanent tissues. So first I will show you all a permanent slide of the xylem tissue. So here I have the xylem tissue, a permanent slide. I will place it on the stage and get it ready to observe. Right. So the specimen is in place and there is enough light. We can observe the tissue. You know that xylem tissue is made up of different types, 4 different types of cells. So when you observe the tissue, try to identify the different types, especially the 4 different types of cells present in it. Now, when I do the adjustment, to a certain extent, you can see that there are different types of cells present in this slide. In the xylem tissue, there are different types of cells. You can observe the different shape and the sizes. So from this observation, you will be able to identify the xylem tissue as a complex permanent tissue. Now we are going to observe the second type of complex permanent tissue, that is the phloem. So in the phloem tissue as well, there are 4 different types of cells. And the most distinct feature is the sieve plate. You can see the polygonal shape and the dots, that is the pores in the cross wall. So from that you can easily identify the tissue as the phloem tissue. So for that again I will get the cell, the slide that has the phloem tissue. This is the permanent slide. I will set it to the correct position. Yes, now there is enough light falling on it. Okay, so we can observe it under low power. And the lens is also low power. So if I look at this tissue, you can see, when I move it around, you can see the sieve plate very clearly. Can you all observe the sieve plate, students? There are 4 different types of cells, but out of those, you can clearly see the sieve plate. So that means this is the phloem tissue. So with this observation and the previous observations, we have observed the different types of simple permanent tissues and the complex permanent tissues. So when you are given a diagram or if you are given a slide to observe and identify, you know how to do that. You have to look for the distinct feature. Okay, students. Now we are going to observe the epithelial tissue using a permanent slide. So you all know the features of epithelial tissue. I will get the permanent slide for the epithelium. Okay, so this is a permanent slide for the epithelial tissue. Before placing the slide, I will put it to low power. Now the slide is in position on the stage and I'll move it so that there is enough light falling on the specimen and the specimen is in place and it is low power, so we can observe it under the low power. When we do a little bit of adjustment, now you can see the epithelial tissue, how the tissue is in place. Okay, students, so you have observed how epithelial tissue looks like and you can understand the features of the epithelial tissue by looking at the slide. Now we are going to observe the blood tissue. You all know it is a type of connective tissue and we will be observing it using the permanent slides available to us. So I will select the tissue, the permanent slide. Okay, I have the permanent slides near here. Okay, so I will place it on the stage and here the light is also on, so there is enough light falling on the sample specimen. And I will move the slide so that the specimen is in the correct position. Now I can adjust it to the low power and we can observe the specimen. So when I do a little bit of adjustment, you can see the blood tissue clearly. You can see the different types of cells present in it. This is under low power, students. So you can observe the different types of cells to a certain extent. To observe it more clearly, I will have to change it to the high power. So now I will adjust the lens to the high power lens. A little more higher. So now you will be able to see the cells more clearly. Can you all observe that there are different types of cells present in this blood tissue? Okay, students. So you have observed there are different types of cells present in the blood tissue. Now we will observe the first type of muscle tissues, that is smooth muscle. So for that I'll be using a permanent slide that is there in the lab. I will get the smooth muscle tissue. Okay. I can observe the features of the smooth muscle using this permanent slide. I will change the lens to the low power. Now we can observe the smooth muscles through this. Okay. Now when I adjust the coarse adjustment, students, you can see the shape of the cell clearly to a certain extent. You can see the shape of the smooth muscle. You can clearly see the spindle shape and the nucleus in the center. So here you can observe all the features of the smooth muscle tissue. Now we are going to observe the skeletal muscle tissue. So here I have a set of permanent slides. From that we can observe how the skeletal muscle tissues and the cells will look like under the light microscope. So I will select the skeletal muscle tissue from this one. I have the slide here. This is a permanent slide that has been prepared for skeletal muscle tissue. Right. Now I have the slide placed on the stage and I will make sure that the specimen is in place. Okay, now it's set, and as you can see there is enough light falling on the stage, so the specimen will be clear to me. First I will observe it through the low power. So the lens is the low power lens. Now if we can observe, under this you can see the muscle fibers and the muscle tissue clearly. So in that you can have a look at the overall features of the striated or skeletal muscle tissue. Okay students, so you have observed all the features of skeletal muscle. Now as I said, we are going to observe a permanent slide of the cardiac muscle tissue. So I will select the slide from the box of slides, the cardiac tissue. Okay, I have it here, so I can place it on the stage, but before that I have to check whether the light is on. Yes, and I'll put it to the low power first. Okay, and also I'll make sure that this specimen is exactly in the correct position so that we can observe it clearly. Okay, now that it's all set and there is enough light falling on the specimen, I can observe it through the low power. So now when I observe it through the low power and I do a little bit of adjustment, yes students, now you all can see the cells of the cardiac muscle clearly. How the cells are, the shape of the cells and the color. Here it is a dyed permanent slide, so you can see it clearly. You can see all the features of the cardiac muscle. Now that is not sufficient, so what I will do is I will change it to the high power. So when we change to the high power, and I'll do a little bit of adjustment. Let me adjust it a little bit. Okay. Now you can see the cells even more clearly. All the features of the cardiac muscles, not all of them, but the features of the cardiac muscle tissue can be seen in this slide. So students, you have observed how the cells look like in a cardiac muscle tissue. Now you all know what we are going to observe. What type of tissue is this? We are going to observe the nervous tissue. How the cells will look like in a nervous tissue. And you all know the basic unit of nerves is the nerve cell or the neuron. So as we did before, I have the set of permanent slides here. So from that, I will take out the nerve tissue. Okay, so this is a permanent slide prepared with nerve tissue. I will place it on the stage of the microscope. Then I will have to, already the light is on, so I have enough light for the specimen. I will have to adjust the slide so that the specimen is visible to me clearly. Okay, now the specimen is ready. First, we will look it through the low power of the microscope. As you can see, already the low power has been selected. So, I'll be using the low power to observe this. Now already the stage is down, I will rotate it a little bit so that I can see the cells somewhat clearly. Now you can see, students, under the low power, you can see the different shape of the nerve cells. These are very much different from the cells that we have been observing so far. You can see the shape of the cell body and the nerve fibers all together present in this slide. Okay, students. So, from this slide, you were able to observe that the cells of the nerve tissue, what we call as the nerve cell or the neuron, is different from the cells that were present in the other tissues.